Okay, so good afternoon, everyone, and welcome to the symposium number 6. This is the radiation-induced cell death, and I'm France Carrier from the Radiation Oncology Department at the University of Maryland in Baltimore. I will chair this session with Dr. Rutul Patel from the Duke University, and we have 4 speakers this afternoon, and we will take a question at the end, right after the 4th speaker. And we'll ask you to use the Q&A box at the end at the bottom of your screen. I have only one slide to kind of summarize our session. So just briefly, as we know, radiation-induced cell death could be mediated by different pathways. We are more familiar with the direct effect of radiation, that is injection of DNA damage, but we're becoming more and more, I think, aware of the indirect and bystander non-targeted effect. And that would be discussed this afternoon by, we have Dr. Stephanie Markovina. She's going to talk about lysosomal induced or mediated cell death. And then we have Dr. Chan, who's going to talk about ferroptosis, oxidation of lipid. We also have effect on the bystander, and the abscopal, but the bystander effect will be discussed by Paul Cahoon. And we have also to keep in mind that the radiation resistance or radio resistance is also something that need to be overcome to increase radiation sensitivity. There are multiple pathways, and Sylvain Farandon will discuss the role of the PI3 kinase pathway in radio resistance. So I will start our session with Dr. Stephanie Markovina. Dr. Markovina is a radiation oncologist at Washington University. She's an assistant professor there, and she did a residency as well at Washington University. She received her MD, PhD from Wisconsin, and she works on gynecology, oncology, thyroid cancer, anal cancer, and brachytherapy. The title of her talk is New Role for Lysosomal Mediated Cell Death in Response to Radiation Therapy in Tumors. So Dr. Markovina, we're ready for you.
Great, thank you so much, Dr. Carrier. Thank you to the organizers, session organizers, meeting organizers for the opportunity to speak today. I'm going to hopefully share my screen here. Okay, hopefully you can all see this. What I'm going to talk about today is a distinct form of cell death that occurs dependent on permeability of the lysosomal membrane. We've termed the cell death mechanism lysoptosis, and we find that it occurs in tumor cells in response to radiation, particularly when we take out a protective protein in the cell. Let's see if I can get my slides to advance here. There we go. Okay. So what I'll hopefully show you today is that SERPINB3, which is a protease inhibitor, resides in the cytoplasm of the cell. And we think its main purpose in being there is to protect cells against the sequelae of lysosomal enzymes when released from the lysosome. So normally the lysosomal membrane is intact, protecting the cell from these highly potent proteases. We find that when we treat cells with ionizing radiation, the lysosomes become leaky, releasing these cathepsin proteases into the cytoplasm. In the presence of SERPINB3, cells are protected against the sequelae of this, but when we take away the function of SERPINB3, we find that tumor cells undergo a necrotic phenotype cell death that we've termed lysoptosis. Now, SERPINB3 is also known as squamous cell carcinoma antigen. It was first discovered or identified in cervix tumor samples back in the 1970s. And since that time, our group and others have shown that high levels of expression of SCCA, both in the tumor and circulating in serum of patients with these cancers, is associated with very poor recurrence-free survival and overall survival and response to treatments like radiation and chemotherapy. Since then, many other groups have shown similar findings in other cancer types, including non-small cell lung cancers, head and neck squamous cancers, hepatocellular carcinomas, esophageal, breast, and anal cancer, all suggesting that high levels of this protein are not only associated with poor outcomes overall, but in many cases in response to anti-cancer therapies like radiation and chemotherapy. So SERPINB3 is a member of the serine protease inhibitor superfamily. It's conserved across all kingdoms of life, including some viruses. there have been greater than 100,000, or sorry, greater than 1,000 serpents have been identified, and they're divided into 16 clades, which describe usually their localization either within or outside of the cell, and as well as their function. And you do know a serpin, whether you realize it or not. Serpin A1 is also known as alpha-1 antitrypsin, which is important for preventing congenital lung disease. Serpin C1 is antithrombin, a critical factor along the clotting cascade. Serpin B14 you may have eaten. It's a protein in the egg and the whites of chicken eggs and polymerizes when heated. So it's responsible for the white substance of chicken eggs. And serpin B3 I'll tell you more about today. That's known as squamous cell carcinoma antigen. Now, initially, squamous cell carcinoma antigen was thought to be one protein, but since we've found that it actually represents two nearly identical isoforms, SCCA1 and 2, or serpin B3 and 4. And their sequence diverges primarily in this C-terminal reactive site loop region, which has two subdomains, including the reactive site loop variable region, which serves as pseudo-substrate for target proteases, and sequence differences in this region result in distinct protease target inhibition. And then the reactive site loop hinge region, which I'll show you in a minute, is important for the structure function of the inhibitory protein function. So in this video, I have a serpin. I did not make this video. It's a very nice depiction of the function of serpins. The serpin molecule has a reactive site loop sticking out of the C-terminus over here, and then a protease will come from the top of the screen. It recognizes this reactive site loop sequence as a substrate. And once it cleaves that substrate, the serpin undergoes a rapid conformational change, removing the protease from that initial cleavage site and the serpin and preventing it from processively cleaving that serpin, thus resulting in a one-to-one and irreversible inhibition mechanism. That's so-called bait and trap inhibitory mechanism. The initial function or importance of this protein was first shown in C. elegans using a model of the serpin B3 ortholog in C. elegans, a protein called SERP6. In this video, which was published back in 2007 by our close collaborators, Dr. Cliff Luke and Dr. Gary Silverman, you'll see a wild type C. elegans animal on the left and a SERP6 null animal on the right, which are dropped into hypotonic conditions, which are can be stressful for the worm. And what you'll see, if I can get this video to play, is that the wild type animal will continue to swim and wriggle happily on the left. Well, we think they're happy anyways, while the SERP6 null animal pretty rapidly assumes this death phenotype or rigid morphology, and then in quite dramatic form will spew its ova through the vulva in what we think is a protective defense mechanism to get their progeny away from whatever the stressor is. So quite a dramatic phenotype in C. elegans. And they found that, again, this results in whole animal death when exposing these animals to hypotonic stress, but it's not unique to hypotonic stress. They found that diverse stressors such as thermal stress, hypoxia, and hyperoxia were the SERP6 null animals were similarly sensitized to those stressors. And the working hypothesis is that SERP6 resides in the cytoplasm of these C. elegans cells, protecting cells against the leaked lysosomal proteases that are released when exposed to stress, and preventing the cells from undergoing necrosis. Thus, when you remove this protective protein, the cells and eventually the animals will die. So we hypothesized that a similar function might be important in human tumor cells and potentially in response to radiation. We placed the sledgehammer cellular stress of radiation therapy at the upstream of this figure, and then think that serpin B3 may be similarly protecting cells against lysosomal leakage in the proteases that are released. So to test this hypothesis, we looked to a panel of cervix cancer cell lines. And here on the left is SW756, which is an HPV positive and P53 wild type tumor cell line that probably represents the majority of human cancers that we see, as well as an HT3 HPV negative and P53 mutant cell line, because we didn't know if there would be an impact on cellular signaling in HPV positive or negative tumors. Treated these cells with increasing doses of radiation to generate a survival curve. And then as a comparison, we added the standard of care radiosensitizer cisplatin, which is used in the clinic to treat patients with concurrent radiation for cervical cancer. which we didn't see impact clonogenic survival, at least very much in either of these cell lines. And that's compared to the single gene knockout using CRISPR-Cas9 of SERPINB3. Significantly sensitized both of these cell backgrounds to ionizing radiation. The further addition of cisplatin to these cells did not seem to have a significant impact beyond the single gene knockout. We've had similar findings in vivo. So when we grow these tumor cell lines in an athymic nude xenograft, flank xenograft model and allow them to grow with sham irradiation, we see that they grow at about the same pace. In contrast, when we treat them with radiation, the HT3 cell line or tumors in mice are actually relatively radioresistant. So those wild type tumors mostly continue to grow. The B3 knockout tumors, in contrast, had a regression in tumor size followed by a sustained tumor growth delay. SW756 tumors are more sensitive in vivo, and thus a single dose of 10 gray resulted in a sustained tumor growth delay in the wild type, but a tumor regression in the SERPINB3 knockout cells. We asked whether this was because of increased cell death. And so turning back to the in vitro model, we treated with increasing doses of radiation and followed these cells for several days after radiation to detect differences in cell death using a cell impermeant dye Cytox. And we find that indeed the SERPINB3 knockout cells are more sensitive to cell death induced by radiation at every dose and at almost every time point. We did not see a big difference in cell cycle distribution or arrest after radiation in these cells. So we didn't think that that was explaining why they were more sensitive. So using DNA content to estimate the cell cycle distribution, both HT3 and SW756, we saw some statistically significant differences, but they were in magnitude fairly small and actually would potentially predict that the B3 knockout cells would be a bit more resistant to radiation given their cell cycle distribution. And following treatment with 4-gray radiation, 48 hours later, we saw the expected G2M arrest in the P53 wild type SW756 cells, which wasn't different between B3 wild type and knockout cells. HT3 cells don't really resort very well probably because they're P53 mutant. And again, we didn't see differences in the B3 knockouts. Similarly, we didn't see a big defect in DNA damage response or repair in the B3 knockout cells. So this is just by measuring phosphorylation of ATM by Western blot. We see both B3 knockout and wild type with induction of phospho-ATM at 30 minutes after 2-gray radiation, both of these backgrounds. And by 24 hours, phospho-ATM is similarly decreased in both the wild type and B3 knockout backgrounds. Using gamma H2AX foci to measure DNA damage, we saw about the same basal level of foci in each of these cell backgrounds. With 2-gray radiation 30 minutes later, we did see induction of gamma H2AX foci by immunofluorescence. We did see a repeatable difference in the short-term number of foci per nucleus in the B3 knockout cells, specifically in the HT3 background, but not in SW756. And although we don't understand that phenotype yet, we're working on it, we do see that even in the HT3 B3 knockout cells, the number of foci returns to baseline in the B3 knockout and wild type cells, suggesting that at least repair appears to be intact. So what is causing cell death then? Well, if we go back to the C. elegans model in the SERP6 wild type and null animals, these are images co-stained for a green fluorescent cathepsin protease substrate that fluoresces when it's cleaved. So it localizes initially to these lysosome-like structures in the C. elegans cells. These are co-stained with a TMR labeled dextran, which also loads into the lysosome. So you're seeing these sort of orangish yellow co-stained vesicles. And what you can hopefully appreciate when we expose these animals to hypotonic stress, again, in this case, in the SERP6 null animals, those lysosome-like structures will disappear and essentially rupture, indicating lysosomal rupture, as compared to the wild type, which largely remain intact. We did a similar experiment with the cervical cancer cell lines and using radiation as the stressor. These experiments were a bit more technically difficult, mostly because of the timeline. It's difficult to follow individual cells and watch for them to die, but we were able to catch a couple. So on the left is the wild type cells, and we have the lysosomes marked in the magenta color using lysotracker deep red. Mitochondria are marked in green with mitotracker, and then propydium iodide is used to detect cell death. And What I'll show you here on the left is one of the wild type cells here, which will become red, indicating death. And you can see that there's still magenta staining in the cytoplasm, as opposed to the SERPINB3 knockout cells here. You'll see one of these where the lysosomes disappear just prior to the cells becoming PI positive. And we have quantitated these using manual tracking of the cells. So using the gold standard for morphology of cell death, electron microscopy, we find no real obvious differences in initial morphology in sham-treated cells in any of these backgrounds compared to their wild-type control. However, when we treat with ionizing radiation and look at the morphology of dead cells about 96 hours later, what we can see is that there really aren't that many dead cells detected in the wild-type control cells. You can see certainly some differences, a lot more activity going on in the cytoplasm here. But in the B3 knockout cells, we saw this pretty dramatic necrotic phenotype of cell death. These cells were all over the place, and we've quantitated them up here. But if we zoom in, we can see not only cell membrane or breaks in the cell membrane and cytoplasmic clearing, but we saw these distinct membrane-bound lysosome-like structures in the cytoplasm, many of which appeared to be ruptured. Using Western blot to detect markers of different cell death pathways, we, looking here with a positive control of etoposide treated HT3 cells, do see end effector caspase cleavage of caspase 3, 7, and PARP cleavage. And that's with only about 15% dead cells at this time point. In the HT3 background with higher doses and later time points of radiation treatment, we can see some evidence of caspase cleavage, and this appears to be a little bit higher in the B3 knockout cells. In the SW756 cells, you can see we really don't detect any caspase cleavage, a little bit of PARP cleavage, which as we know is not specific for apoptosis, but otherwise doesn't appear to be a large proportion of cell death that's induced. Similarly, if we look at Gasdermin D cleavage, which is a marker of pyroptotic cell death, positive control band is shown here in this third lane of the central positive control lanes, and we really were unable to detect Gasdermin D cleavage in any of the radiated samples. Similarly, markers of necroptosis, including phosphorylation of MLKL and phosphorylation of RIP3 and RIP1 kinases, were detected in our positive controls, but again, really not detected in any of the samples treated with increasing doses or time points after radiation. Inhibitors, pharmacologic inhibitors of each of these cell death mechanisms showed similar results. We used pan-caspase inhibitor, QVD-OPH, necrostatin 1, ferrostatin, and this is a caspase-1 inhibitor, which should inhibit pyroptotic cell death. In the SW756 background, didn't inhibit any radiation-induced death in the B3 knockout cells. However, in the HT3s, the pan-caspase inhibitor did partially inhibit cell death in both the wild type and the B3 knockout cells, consistent with our Western blot results. So this suggests that at least some caspase-dependent cell death might be occurring. And when we looked closer at apoptosis to see if the machinery were intact in these cell backgrounds to induce apoptosis, we did indeed in the wild type cells see classic apoptotic morphology using electron microscopy. The B3 knockout cells also died. However, cell death morphology in these cells was actually necrotic. And that was those findings were consistent with what we saw using pharmacologic inhibitors of cell death. So the DVD-CHO, a pan-caspase inhibitor, did inhibit wild type induced cell death. However, it did not inhibit cell death induced by storage-borne B3 knockout cells. In contrast, a cysteine protease inhibitor, E64, did inhibit cell death in the B3 knockout cells, suggesting that cathepsin protease activity is important for mediating cell death in response to this classic apoptosis inducer. We wanted to know if lysosomal permeabilization was occurring in these cells, and so we used a system where we used 4 different molecular weight dextrans labeled with distinct fluorophores, and could thus measure their leakage from the lysosomal compartment using fluorescent microscopy. And so here are the untreated or vehicle-treated cells, wild type on the left, B3 knockout on the right. We can see that there's maybe some leakage over time, but largely these signals are staying intact in the lysosome. The wild type treated cells did have some leakage of the smallest molecular weight dextran, the 3 kilodalton, suggesting that there is some impact on permeability of the lysosomal membrane. But all these larger dextrans, including just the 10 kilodalton, remained within the lysosome, at least until later time points. This is in contrast to the B3 knockout cells, which had a pretty rapid loss of all of the molecular weight dextrans, consistent with loss of integrity of the lysosomal membrane. This occurred also when we treated with the MTP-dependent necrosis inducer. Sorry, this is hydrogen peroxide. Again, we see what we would expect by electron microscopy in the wild type cells with abnormal morphology mitochondria and these dying cells. Similarly, we saw that effect in the B3 knockout cells, but also saw this dramatic necrotic morphology as well. Inhibitor of MTP-dependent necrosis, cyclosporin A, inhibited cell death in the wild type blue bars here, but did not inhibit cell death in B3 knockout cells. And again, E64D, that cysteine protease inhibitor, was able to inhibit at least partially cell death in these B3 knockout cells treated with hydrogen peroxide. The same assay to look at lysosomal membrane permeabilization showed a similar finding in the wild type cells with some leakage of the smallest molecular weight dextran, but more leakage of all of the dextrans in the B3 knockout cells, suggesting that lysosomal integrity was more compromised in the absence of serpin B3. This happened again when we treated cells with necroptosis inducing conditions, TNF alpha, pan-caspase inhibition, a SMAC mimetic, and cyclohexamide was required to induce death in the wild type cells, where we saw classic expected morphology of necrosis. And in the wild type cells, again, these very large cytoplasmic membrane bound inclusions, which were consistent with lysosomes. Necrostatin 1 inhibited completely cell death in the wild type cells and partially in the serpin B3 knockout cells, but E64D was able to protect cells in the B3 knockout setting as well. And again, similar findings with leakage of the lysosome where the dextrans were largely intact in the B3 wild type cells and leaked pretty quickly out of the B3 knockout cells. This wasn't true for every inducing pathway. So here, when we treated cells with Erastin, which classically induces ferroptotic cell death, we saw similar findings with inclusions in the mitochondria, really abnormally appearing mitochondria. We also saw a pretty consistent cell death in both the wild type and the B3 knockout cells that was inhibited by addition of the inhibitor ferrostatin 1. You can see here both. And also to support this, we did, although there was some leakage of lysosomal dextrans in the B3 knockout cells, it wasn't that dramatic an early decrease that we saw in the other inducers. So what this suggests to us is that serpin B3 is important for maintaining the integrity or protecting the cell against these lysosomal cathepsins from diverse stimuli that might induce lysosomal leakiness. But what we wanted to know is in response to radiation, is the protease inhibitory function required? Are we seeing really truly the same function in response to radiation? So we generated a single point mutant within the hinge region of the reactive site loop, which prevents this conformational change and therefore prevents the ability of the serpin to effectively inhibit target proteases. And we expressed in cell line, cervix cancer cell lines that don't normally express serpin B3, either an empty vector control, the wild type serpin B3 protein, or this point mutant to see if we could rescue this phenotype with wild type or point mutant, hinge region point mutant. And what we saw is that we could, in fact, protect cells against radiation by clonogenic survival and proliferation assay by overexpressing the wild type serpin B3. In contrast, the point mutant did not protect cells against radiation in either of these assays. Similarly, in vivo, we saw that expression of B3 promoted appeared to promote more tumor growth in unirradiated tumors using the C33A background, whereas the point mutant did not have that effect, similar to vector control. When we irradiated these tumors, the B3 wild type expressing tumors were more radioresistant than either the vector control expressing or the point mutant, suggesting that the reactive site loop and protease inhibitory function of serpin B3 is required for this cytoprotection. To understand what downstream proteases might be mediating this cell death mechanism in response to radiation, we looked to our primary tumor specimens. So RNA-seq analysis of 66 primary pre-treatment tumor specimens from cervix cancer patients is shown here. And we looked particularly at the cysteine cathepsin proteases. The most highly expressed were cathepsins B and H, which actually have more exopeptidase activity inside the cell, which would not be inhibited by serpin-B3. Serpin-B3, because it's cleaved in the center of the protein, requires more endoprotease activity and therefore inhibits endopeptidases. And so we can see overall pretty low expression of these proteins by RNA-seq. However, if we blow this up, we see that L, S, and K are really the ones that have any level of expression in cervix tumor samples. In the cell lines, we didn't find any detectable intracellular cathepsin S or cathepsin A. There was in the wild type serpin-B3 cells a good amount of mature cathepsin L. And interestingly, in the serpin-B3 knockout cells, that basal level is actually significantly downregulated, potentially because cells that did not have inhibition of cathepsin L were dying. And we generated double knockouts, both single knockout and double knockout in the serpin-B3 wild type and serpin-B3 knockout background of cathepsin L, and treated these cells with radiation and found that the cathepsin L knockouts in the wild type serpin-B3 wild type background did not appear to impact response to or cell death following radiation, but were at least partially protective in the serpin-B3 knockout cells, suggesting that cathepsin L is at least one of the downstream executing proteases involved in the cell death pathway. So with that, our key findings so far that serpin-B3 protects cervix cancer cells against radiation-induced lysosome-dependent death that follows by a necrotic morphology. And we think this is lysoptosis-like death. Cathepsin L is at least one of the key proteases involved in executing lysoptosis in cancer cells. And that loss of the intracellular serpin not only permitted lysoptosis in response to radiation, but appears to sensitize cells via a lysoptosis-like death to diverse cytotoxic stimuli, including other chemotherapeutic agents. Unanswered questions, there are many things that we're focusing on currently in the lab. We'd like to know how radiation induces lysosomal membrane permeabilization. Is this a direct or indirect effect or a combination of many upstream factors? What other factors can protect against lysoptosis and or stabilize or destabilize the lysosomal membrane? Again, getting at how to therapeutically target this. And based on what I've shown you, are serpin-B3 and lysosomal destabilization potentially therapeutic vulnerabilities that we can target for radiosensitization? With that, I have a lot of people to thank, namely my lab members who did the bulk of this work. I'll give a shout out to 2 of my trainees who have poster sessions at Radiation Research this year. Some very key collaborators and friends, Cliff Luke and Gary Silverman, and my mentors with whom I wouldn't be here, of course, Stuart Kornfeld, Perry Grigsby, and Julie Schwartz, to name a few of the most important. Also, thanks to my funding bodies. And with that, I thank you for your attention.
Thank you, Dr. Markovina. I think we'll have to wait till the end to ask questions, but we have a lot to chew on. I mean, it's really interesting, and I can't see how we can really roll this talk in our next talk on ferroptosis, lipid oxidation. Okay, let's just put it this way. I think there's a lot, of overlap and crosstalk between your observation and our next speaker. So our next speaker is Dr. Simon Chang. And Dr. Chen is a radiation oncologist. He's also an assistant professor at Columbia University. He received his MD PhD from New York University and did his radiation oncology residency there as well. He's currently the director of the residency program in radiation oncology at Columbia. And the title of his talk is ferroptosis. So Dr. Chen.
Hi, I'm Simon Chang. I'm Department of Radiation Oncology at Columbia University. I'd like to thank the organizers of this panel and Radiation Research Society for inviting me to talk about radiation-induced ferroptosis. I have nothing to disclose. Classically, radiation was thought to mostly induce DNA damage, which results in cell death, such as apoptosis. However, it is now known that radiation could cause other forms of regular cell death, including necroptosis, autophagy, and senescence. In addition, radiation induces other cellular damage, such as protein oxidation, lipid peroxidation, and induction of inflammatory immune responses, which have been shown to greatly enhance tumor control. One other newer form of regulated cell death is called ferroptosis, which is death by overwhelming lipid peroxidation. Although it's been known that oxidative stresses and reactive oxygen species can cause cellular lipid damage, leading to cell death, the regulatory pathways for ferroptosis were not identified until small molecule inhibitors of the antioxidant pathways were found. Brent Stockwell, our collaborator at Columbia, identified AKE, which inhibits cysteine XC, which is a cysteine glutamate transporter, which brings in cysteine into the cell. And cysteine is a key essential factor in production of glutathione. Glutathione, in return, is a central cofactor of GPX4, which reduces and repairs lipid peroxidation. Other small molecules, such as RRS3, were found to inhibit GPX4 and induce ferroptosis. Oxidative polyunsaturated fatty acids, or PUFAs, was shown to be the key molecules in inducing ferroptosis. And antioxidants that are lipophilic, that bind to the cell membranes or plasma membranes, such as vitamin E and ferrostatin, was shown to be able to rescue the lipid peroxidation and prevent ferroptosis. There are certain key activators of this pathway also that's been known. For example, transferrin receptor, which brings in iron into the cell, which has caused its free iron labile. This free iron labile is able to enhance lipid peroxidation by increasing Fenton reactions. Other enzymes, such as ASCL4, can increase PUFA levels, which is needed for lipid peroxidation and ferroptosis, driving cell death. So what does cells undergoing ferroptosis look like? So this is a time-lapse video by by Brent Overgaard at Memorial, and he shows here a time-lapse video of a tumor cell undergoing ferroptosis. What we see is cell swelling, which is noted by the red dye. And then there's influx of calcium, and then a breakdown of the cell membrane, which bursts the cell and causes the cell to be labeled by cytotoxic screen. So this compares to death by apoptosis, which is different because that causes cell shrinkage and has intact cell membranes. So radiation is known to cause direct damage to DNA and to lipids and proteins, but it also could cause indirect damage through causing reactive oxygen species that further damages the proteins and lipids and DNA. So we had a question to see if radiation could cause lipid peroxidation and ferroptosis and synergize with ferroptosis inducers. So we first tested if radiation could synergize with ferroptosis induces IKE and RSL3 in inducing cell death. So in this clonogenic assay of HE1080 cells, which are fibrosarcoma cells, which are ferroptosis sensitive, we showed that there is synergy of cell killing between ferroptosis induces IKE and RSL3 together with radiation. We then wanted to see what was the predominant form of cell death caused by radiation in HT1080 cells. So we tested ferrostatin, which is lipophilic antioxidant, to see if it can inhibit ferroptosis and rescue radiation cell death in HE1080 cells, and it does. Interestingly, we also tested ZVAD, which inhibits apoptosis, and to see if it could actually rescue cell death caused by radiation in these HE1080 cells. And there was no cell rescue with ZVAD. This suggests that certain cells in certain contexts, such as in HE1080 cells, ferroptosis can be the predominant form of radiation-induced cell death. We then wanted to see how common was radiation-induced cell death and apoptosis in other cell types. So we tested the synergy of lung cancer cells such as A549 cells or PC9 cells and gliomas, U87 cells, and other sarcoma cells, and to see if there was a synergy between radiation and apoptosis inducers. In all the cell types we tested, there was synergy to some degree or another with radiation and apoptosis inducers. This suggests that this phenomenon of radiation-induced cell death by apoptosis is a common phenomenon that can be associated with cell death in many cell types. The next thing we wanted to see was that what was the potential mechanism of radiation apoptosis induced and radiation synergizing together and causing cell death. So first, we wanted to see if there was a synergy between radiation and apoptosis inducers in causing, altering, or producing or repairing DNA damage. We looked at first gamma H2X, which labels double-stranded DNA breaks. We first looked at apoptosis inducers to see if they could themselves cause DNA damage with gamma H2X, which they don't. We looked at then 6-gray radiation to see if it causes gamma H2X at 30 minutes, which it does, and seeing if the addition of IKE and RSL3 can enhance the DNA damage caused with gamma H2X, which we did not see. We further looked at ferrostatin, which inhibits apoptosis, to see if there was any effects on DNA damage caused by radiation. And there was no altered effects with the addition of ferrostatin plus radiation. We also looked at 6 hours post-radiation to determine if there was any altered or delayed DNA repair, and there was also no differences between different groups. This suggested that radiation and apoptosis inducers did not enhance DNA damage. We further then looked at to see if liver peroxidation was a possible mechanism between for the synergy. So we combined apoptosis inducers and radiation to see if it increases liver peroxidation. So this is a T-bar assay which measures MDA, which is one of the more toxic byproducts of liver peroxidation. Radiation IKE by themselves slightly increases MDA levels, but the combination of IKE and radiation significantly enhances MDA liver peroxidation. The addition of ferrostatin prevents the MDA production in the radiation and combination of radiation plus IKE groups. We then looked at untargeted lipidomics analysis to see what was the lipid species that would form with IKE and radiation synergy. And what we show is that both IKE and radiation actually have the same profile of lipid peroxidation. And these include several lysophospholipid, which are derivatives of oxidative PUFAs. And this suggests that these isophospholipids can be potentially specific biomarkers for radiation and IKE-induced apoptosis. So at Columbia, at RARAF, the Radiological Research Accelerator Facility at the Center for Radiologic Research, we have a proton microbeam that's capable of delivering precision and automated subcellular radiation to microsubcellular compartments, such as shooting a microbeam through the nucleus only, or through the cytoplasm and some membrane only. So what we decided to do was use the microbeam to see if there was a difference in subcellular radiation and the synergy between apoptosis inducers and and radiation. So what we looked at was we compared cytoplasmic and nuclear radiation in inducing ferroptosis. IKE and RSL3 had no or very little synergy when combined with nuclear radiation. And we confirmed that nuclear radiation did induce gamma H2X double-stranded DNA breaks, while cytoplasmic radiation had very little gamma H2X expression. However, when we combined cytoplasmic radiation with, there was significant synergy between cytoplasmic radiation and IKE and RSL3. Looking at gamma H, for H&E, there was which is a marker for lipid peroxidation. Only cytoplasmic radiation increased gamma for H&E, and nuclear radiation had very low for H&E. The next thing we wanted to do was to confirm if this synergy occurs in vivo. So we looked at to see if ferroptosis induces and radiation can improve tumor controls of a mouse model of the fibrosarcoma model, flank model. The combined group basically had no growth of tumor within the 2 weeks analyzed compared to the single treatment of IKE or radiation alone, suggesting significant synergy of IKE and radiation. We also looked at another human model, a dual human lung cancer model, PDX model, and confirmed that the combination of radiation and IKE has a significant synergy in vivo. We also looked at another inducer of ferroptosis, sorafenib, which inhibits systemic C, the cysteine transporter, and that also shows enhanced tumor control with radiation in both the human sarcoma flank model and the PDX lung cancer model. So this suggests that ferroptosis induces and radiation can act in vivo in controlling tumors synergistically. So the question now becomes, what is the mechanism causing ferroptosis? It could be that the radiation is causing direct DNA damage or indirect DNA damage by the reactive oxygen species, damaging the lipid membrane by itself. However, given that now there's a very well-known regulatory network of proteins and transporters and enzymes that could regulate ferroptosis, we wanted to see if radiation could affect the oxidative pathways, such as regulating glutathione, in looking at and glutathione is known to activate GPX4. So what we did was we wanted to see if glutathione levels was altered when we combined radiation plus ferroptosis inducers. So in this assay for oxidative glutathione, we showed that both IKE and radiation induces glutathione levels. And there was a synergistic reduction of glutathione with the combination of both IKE plus radiation. This suggests that radiation can act further upstream in the pathway more than at the level of lipid peroxidation. So is glutathione the only radiation mechanism for induced ferroptosis? Likely not. Other groups have already shown that upregulation of other enzymes such as ACL4 by radiation promotes ferroptosis, and that this is a potential synergistic mechanism. ASCL4 makes PUFAs in the cell membranes, which is oxidized in ferroptosis. And actually, there's other mechanisms that people have proposed, like altering systemic C levels by radiation already. So what cell death pathways do radiation activate? Bo Yi-Gan and his group at MD Anderson have looked into radiation-induced ferroptosis, and they tested various cell death inhibitors with radiation to see if there was what various types of radiation-induced cell death mechanisms were activated in different tumor types. Here you see three different tumor types, and it seems like radiation can upregulate several different cell death pathways at the same time in the same population of cells, to varying degrees according to different tumor types. So it's unclear what are the regulatory mechanisms that controls the switching between cell death mechanisms induced by radiation. Versus, does the cells go between apoptosis or ferroptosis? And what is the switch between, for example, these different cell death pathways is not known yet. And obviously this will be an area of focus and active research. So I'd like to thank my members of my lab, members of Brent Stockwell's lab, Ying Li, especially for doing this radiation-induced ferroptosis work, people and Lisa Kachnik, my chair at Columbia in Radiation Oncology, and our funding mechanisms, Barry Stockwell and Longevity Foundation. Thank you.
Thank you, Dr. Chan. We will move right along. I think I'm going to hand over the chair to Dr. Patel, who's going to introduce the next two speakers. And at the end, like I mentioned at the beginning, at the end of those 4 talks, then we'll open the session for question and answer. Please use the QA box at the bottom of your screen. Thank you.
Thank you, Dr. Kerr. It's my pleasure to introduce our next speaker, Dr. Sylvain Perdon. He's a research scientist at Ohio State University. He specializes in cellular and molecular biology, especially in telomerase physiology, stem cell biology, identification of biomarkers, and preclinical development of anti-cancer drugs. Title of his talk is CoA synthase, a novel predictive marker and functional protein that mediates radiation resistance via PI3 kinase signaling pathway in rectal cancer. Dr. Perdon, floor is yours.
Today, I will present you our work on the identification of CoA gene expression as a novel predictive biomarker of radioresistance in rectal cancer. The standard treatment for advanced rectal cancer is based on a neoadjuvant chemoradiation followed by surgery and then an adjuvant chemotherapy. But despite this therapeutic treatment, there is a lot of heterogeneity in patient response. The American Joint Committee on Cancer developed a scoring system to categorize the heterogeneity based on the tumor regression. It goes from score 0, where there is no viable tumor cells left after treatment. These patients are considered as complete responders, to score 3, where there is only a minimal or no tumor kill. In 2015, Dr. Adam Mays from our group showed that the AJCC score is a powerful prognosis marker in rectal cancer. However, the AJCC score can only be determined after treatment. So the question we are trying to answer here is how to predict those patients. For that, there is a need of predictive biomarker. To answer this question, we used the following approach. We recruited 33 patients with rectal adenocarcinoma. From these patients, we harvested pre-treatment biopsies and analyzed their transcriptomic profile using microarray analysis. In the meantime, the patients received their neoadjuvant chemoradiation, and by the time of their surgery, pathologists determined their AJCC scores. We then compared the transcriptomic profile to the AJCC score. All 4 AJCC groups were compared for differentially expressed genes using ANOVA test, and 145 genes were isolated. We then compared the complete responders versus the other groups and isolated 170 genes. Finally, we compared the worst responders, AJCC3, with all the other groups and isolated 218 differentially expressed genes. When we cross-reference these 3 lists of genes, only 3 genes are shared. One of them is CoA, encodes for the coenzyme A synthase. This enzyme catalyzes the two last steps of the coenzyme A synthesis. We first validated our microarray results by RT-qPCR on the 33 biopsies as well as normal mucosa. Using this technique, we observed a stepwise increase of COSY expression with increasing AJCC score. Interestingly, we also found that COSY expression was significantly higher in rectal cancer tissue compared to normal tissue, and this finding was supported by similar results found in 3 independent cohorts from Oncomine. To further evaluate if our findings were reproducible in other cohorts, we tested the ability of COSY expression to predict therapeutic response of rectal cancer by using the microarray dataset from Dr. Watanabe, published in Cancer Research in 2009. His study didn't bring the granularity between the AJCC score only between responders and non-responders. Therefore, as a comparison, we compared AJCC 0 and 1 for responders patients to AJCC 2 and 3 for non-responder patients. Both cohorts showed a significant overexpression of COSY in the non-responder patients. The ROC analysis estimated an area under the curve of almost 70% for our cohort and 77% for Dr. Watanabe's cohort, demonstrating a good capacity of COSY expression to distinguish responder patients from non-responder patients. Taking together these data indicate that COSY expression is a potential predictive biomarker of patient response. Considering the correlation of COSY expression and radiation response in the clinical setting, we investigated COSY expression in multiple colorectal cancer cell lines to expand the study in vitro. COSY expression was measured using RT-qPCR in 7 cell lines, and on these same cell lines we also measured by clonogenic survival assay the radiobiologic index D10, which corresponds to the dose in gray required to kill 90% of the cells. By comparing both parameters, we observed a significant positive linear correlation. Next, we tested if COSY expression knockdown itself could alter the radiation response. For that, we used the 2 cell lines with the strongest expression of COSY and generated stable COSY knockdown using 2 different shRNAs. We then submitted these cell lines to 4 or 6 grays of irradiation and quantified the percentage of cell deaths by annexin 5-PI 72 hours after irradiation. For both cell lines and both knockdowns, we found a significant increase of cell deaths after irradiation. The same observation were made looking at the clonogenic survival assay, where both knockdowns were significantly lower than the non-targeting shRNA control cell line. We then conclude that COSY itself influences the radioresistance in vitro. We then created an in vivo model in which we injected subcutaneously the RKO control cell lines and the 2 COSY knockdowns in different mouse groups. Once the tumor reached 200 cubic millimeters, the mice were randomized between control and irradiated group. The irradiated group received fractionated irradiation of 2 grays per day during 5 consecutive days. As you can observe on the left panel, we did not see any differences in tumor growth between the groups without treatment. However, in the irradiated group, the tumor from the 2 COSY knockdown lines exhibited a significantly slower growth compared to the non-targeted shRNA, suggesting that COSY knockdowns enhances the effect of radiation and impede tumor growth, but does not affect the non-treated tumor growth. 21 days after the beginning of irradiation, we harvested the tumor and stained slides for proliferation marker Ki67 and apoptotic marker cleaved caspase-3. The non-targeting shRNA tumor demonstrated extensive proliferation and minimal apoptosis compared to the COSY knockdowns. So in summary, we have shown that COSY can alter radiation response in vitro, in vivo, as well as in patients. Our next question was to understand the mechanism beyond the COSY-mediated radioresistance. For that, we used the microarray data from our cohort and compared the gene expression of the bottom 25% of patients for COSY expression, here named COSY low, to the top 25% for COSY expression, here COSY high, in order to identify associated genes or pathways. We identify 660 differentially expressed genes between these two groups. To understand which class of genes are associated with COSY, we submitted this list of genes to a gene ontology analysis. Only 4 GO were upregulated in the COSY high group. The most significant one is named response to oxidative stress. This analysis was then completed by a gene set enrichment assay, and among the reactome gene set significantly enriched, we highlighted PI3K-AKT activation pathway, EGFR signaling in cancer, and PDGF signaling. All these pathways are known to be activated by irradiation and triggers the activation of the downstream PI3K-AKT pathway. At this point, we hypothesized that COSY could modulate the PI3K-AKT pathway activation after irradiation in cancer. Using liquid chromatography and mass spec, we identified a physical interaction between COSY protein and P85 alpha, which is the regulatory subunit of PI3K. As an additional evidence of their interaction, we also observed a partial overlap of their localization using immunofluorescence. To determine if COSY modulates the radiation-induced activation of PI3K pathway, phospho-AKT and phospho-mTOR, the downstream effector, were measured in stable COSY knockdown and control condition 24 hours after irradiation. We also generated 2 COSY overexpression cell lines. For RKO and HRT18, we found a lower level of phospho-AKT and phospho-mTOR in their 2 knockdown compared to their control condition. Conversely, when COSY is overexpressed, phospho-AKT and phospho-mTOR were strongly increased compared to control. The PI3K-AKT-mTOR pathway is known to directly regulate the DNA damage repair machinery. The role of COSY in DNA damage repair was assessed by quantifying the residual DNA damages 24 hours after irradiation by immunostaining of gamma H2AX. In both cell lines, there was a significant increase of residual DNA damages in the shRNA COSY lines compared to control. To understand the dynamic of DNA repair, we then investigated the expression of DNA repair proteins. Firstly, and interestingly, the level of COSY doesn't seem to be altered by irradiation. On the left, the RKO cell line, only the level of MRE11 seemed to be altered by the COSY expression. However, the HRT18 cell lines, which is an homologous recombination deficient cell lines, displayed a strong alteration of DNA PKCS expression that directly correlates with COSY expression after irradiation. As a general summary, we have discovered that COSY has a strong predictive values for tumor resistance to neoadjuvant chemoradiotherapy. We have shown that COSY is differentially expressed among AJCC tumor regression score and promotes resistance in vitro and in vivo. Mechanistically, we found that COSY modulated the PI3K-AKT pathway after irradiation, which finally modulates the DNA repair. I would like to acknowledge all my coworkers who helped the project, as well as the foundation that provided fundings. Thank you very much.
Thank you, Dr. Ferron, for a wonderful talk. We will wait till the end for the questions. Our next speaker is Paul Cahoon. He's a PhD student based at Queen's University, Patrick G. Johnson Center for Cancer Research. And the title of his talk is Investigating the Role of the C-gazetine Pathway in Mediating Radiation-Induced Bystander Effects. Floor is yours, Paul.
Hello everyone, my presentation is titled Investigating the Role of the C-gazetine Pathway in Mediating Radiation-Induced Bystander Effects. I'm going to start my presentation by giving a quick background on the direct and the indirect effects of radiation. So the conventional understanding of radiotherapy involves ionizing radiation, traversing the nucleus of a cell, damaging its DNA, and causing cell death. However, the relationship between radiation and cell death is actually known to be more complex than this, and that's due to further associated indirect effects. One example of such an indirect effect is the radiation-induced bystander effect, and this describes the observation of unirradiated cells responding to their irradiated neighbors. A schematic illustration of this is shown on the right. So if you imagine a setup where part of the cells in the population are directly irradiated, then you'd see cell death in the irradiated cells as expected, but you'd also see heightened levels of cell death in the out-of-field cells. and that's due to so-called bystander signals that are being released. Research of the bystander effect dates back decades, and although many different mediators have been suggested, the exact mechanisms underlying the bystander response are not known. Another example of an indirect radiation effect is the abscopal response, and this refers to the phenomenon where, following the irradiation of a primary tumor, the immune system is provoked, and regression is observed at a distant unirradiated site. The abscopal response, along with bystander effects, both contribute to cell killing following radiation exposure, and my research aims to investigate if these two seemingly distinct processes share a common underlying mechanism. In particular, my research is focused on the cGAS sting pathway, a DNA sensing pathway normally associated with immune response. Results have indicated that this pathway could play an important role in mediating radiation-induced bystander effects. A rough pathway diagram for cGAS sting is shown on the right of the slide. So when DNA is damaged, small fragments of double-strand DNA can break off and end up in the cytoplasm. cGAS detects these double-strand DNA fragments, and this leads to an inflammatory response caused by downstream production of type 1 interferons and other inflammatory cytokines. In order to study the role of cGAS sting and bystander effects, I initially utilized 3 MIGCAP-derived mouse prostate cancer cell lines, an unedited parental line, and 2 further knockdown lines where the cGAS gene and the sting gene were knocked out using CRISPR. I studied the bystander effect by performing partial field exposures and measuring the clonogenic survival observed in the out-of-field regions. When the MIGCAP cell lines are exposed to uniform fields, they exhibit the expected linear quadratic behavior. However, measuring the out-of-field survival shows there is a difference in the strength of the bystander response under each of the conditions. Under normal conditions, when you deliver a half-field exposure to cells and measure the out-of-field clonogenic survival, you'll see something like the blue line in the bottom right-hand figure, where, due to bystander signaling, you'll observe lower than expected out-of-field survival. But when the cGAS sting pathway was interrupted, either through gene knockout or by adding a DNase enzyme immediately after irradiation to degrade any extracellular DNA, we found that the bystander response was mitigated. This response is not exclusive to MIGCAPs. Additional experiments also verified this behavior in MDA-MB231 human breast cancer cells and MCF10A normal epithelial cells. The out-of-field response of each of these cell lines is shown in the right, where CRISPR knockdown of the sting gene and addition of DNase enzyme once again mitigated the bystander response. Western blotting confirmed that all cell lines mentioned were signaling as expected. To investigate this behavior further, I utilized co-culture assays. When carrying out the experiments, I didn't use a setup like the one pictured in the bottom left-hand diagram, but the schematic is useful for describing how the assay functions. In this setup, you culture 2 different cell lines, parental and sting knockout, alongside each other. One cell type is directly irradiated and acts as the in-field cell, transmitting the bystander signal, whereas the other acts as the out-of-field cell and receives the bystander signal. The survival trends resulting from these assays are shown and indicate that the pathway status of the out-of-field cell is what drives the bystander response. Specifically, if the out-of-field cell has the cGAS sting pathway interrupted through gene knockout, we don't see a bystander effect, regardless of the characteristics of the in-field cell. And when the out-of-field cell has a cGAS sting pathway intact, the opposite is true. The influence of the cGAS sting pathway in mediating the bystander response can also be studied by adding DNase to cells at different time points post-irradiation. DNase has the effect of degrading any extracellular DNA and thus removing the activator of the cGAS sting pathway. When DNase is present in cells at earlier sub-24-hour time points, the bystander response is mitigated as before. However, when DNase is added at later time points post-irradiation, the survival trends show that this is insufficient for mitigating bystander effects. This shows that whatever is driving the bystander response appears to be taking effect prior to 24 hours. Another area we're researching is the effect of activating and inhibiting various pathways associated with cGAS sting. So, for example, the RF3 and NF-kappa B pathways are co-activated alongside cGAS sting and also have inflammatory endpoints. The result of inhibiting the NF-kappa B pathway is shown in the right-hand figure, where there is no significant difference in out-of-field survival between the untreated and the drugged cells. This suggests that whatever is causing the toxicity associated with the bystander response is not a byproduct of NF-kappa B. The effects of adding G10 sting activator were also tested. The intention of adding a sting agonist was to try and induce a bystander response in unirradiated cells, where parental cells would be more sensitive than their sting knockdown counterparts. However, preliminary results show similar levels of survival for parental and sting knockout cells in response to the drug. Perhaps this suggests that while sting is a necessary component of the bystander effect, it is only one facet of the response. The data presented today shows how the CGAS sting pathway plays an important role in mediating bystander effects. And together, these results have led to a hypothesis where, following irradiation exposure, double-strand DNA is released by directly irradiated cells into the media where it can be taken up by bystander cells. The subsequent increase in double-strand DNA within the inner out-of-field cells leads to activation of the CGAS sting pathway. The endpoint of this pathway involves production of inflammatory cytokines, causing an inflammatory response within cells. This inflammatory response leads to genotoxic stress and an increase in cell death within bystander cells, contributing to the phenomenon of radiation-induced bystander effects. In terms of future directions to test this hypothesis, I'm planning to perform additional Western blots to analyze CGAS and sting expression under various irradiation conditions. I'm also in the process of obtaining data quantifying the amount of double-strand DNA present within cells at various time points post-radiation, and I'm doing this by performing immunofluorescent staining for double-strand DNA. I intend to quantify the amount of double-strand DNA present in the media at various time points too, and additionally, investigating the effects of an RF3 inhibitor on radiation-induced bystander effects could also be of value. Thanks everyone for listening to my presentation. Here are my acknowledgements, and let me know if you have any questions. Thank you.
Thank you, Paul. Dr. Currier, you want to take it away with the Q&A session?
Yes, we can open now the session. I'm not seeing any, I'm just seeing only one question in the box, and I believe it was for our first speaker, right? They want to know if the process that were discussed are dependent on the level of oxygen, and then in the context that whether some of the changes found with flash could occur due to the local change on oxygen impacting this process that modify radiation response. That was a question from Errol Schwartz. I mean, if Errol is still there, you can take the floor and open your mic to clarify your question. Yes, no?
I guess I'll go ahead and answer, and then if you have any follow-up questions, happy to answer those. So although we haven't directly tested that in hypoxic conditions, we do know that SERPIN-B3 expression seems to be induced in response to hypoxia, potentially via the HIF transcription factors. And I showed you some of the C. elegans data showing that the SERP6 knockout, that's the ortholog of SERPIN-B3, are sensitized to hypoxic conditions by whole animal death. So I think it would suggest that these pathways might also be intact in hypoxic conditions and not entirely dependent on high levels of oxygen, although we haven't, like I said, tested that in our cell lines yet. With respect to flash, we are hoping to look at the effects of flash on the lysosomal membrane integrity, both with and without SERPIN-B3 in the cell. And our clinical department, physics department are working on adapting one of the proton therapy machines to be able to test that in our department. So hopefully coming soon.
I have a couple of questions of my own. When you talk about the SWS756 cells, that they are P53 wild type, they are also HPV positive. So that means that P53 is not going to be active, right? It's going to be deactivated through E6. So basically, that's why there's not much difference between the two cell lines, between the mutant and the wild type, because the wild type doesn't have really an active P53. So my question to you, did you look at HPV negative cells that would be P53 wild type, and then if there you see any differences?
So all of the available cervix cancer cell lines that we have access to, there are a number of HPV negative cell types, and they're all P53 mutant. So we haven't gone back to knock in wild type P53 in those cells and see if we see a dramatic difference. We didn't have a specific hypothesis to why the phenotype would be different, either of those cell types, but wanted to test it, given that cervix cancer can present in both ways. Really, the only difference we saw was in checkpoint resorting after radiation in the SW cells.
Then the other question I have for you is that one of the main components of the lysosomal membrane is the single myelin, this lipid, and that would bring into the next talk with Dr. Chen with lipid oxidation. but also with the concept that this sphygmomyelin, will go to ceramide and then you're going to go to apoptosis. So that's why if you have increased activation of this sphygmomyelin, that why apoptosis would not be then the endpoint as a result.
Yeah, I guess we don't know necessarily. We're looking at what the signaling is, I guess, at this point, both upstream and downstream factors. Now, when we measure leakiness of the lysosome after radiation, we see a small amount, but it appears to be, I don't know if you remember from the videos, that there's sort of a threshold that's passed, and then the lysosome ruptures and releases all these proteases into the cytoplasm, and then the cell dies pretty quickly after that. So we don't know what other signaling might be involved prior to that, but it seems like that threshold event probably trumps any other signaling and pushes the cells down a necrotic death pathway. We haven't looked in the SERPINB3 knockout cells whether that impacts sphingomyelinase or ceramide production.
Do we have any more questions for Dr. Markovina?
Can I ask a question to Dr. Chang?
Yes.
Sure.
Yeah, so you showed that post-radiation, the glutathione synthesis decrease. Do you think that inhibition of glutaminase will further increase the response? Like, how would you see any inhibition of glutaminolysis process directly feed into the radiosensitization part?
Yeah, I think that's a very good question. Yeah, so I think potentially, yeah, with glutaminases, inhibition with apoptotic inhibition may sort of increase the sensitization. I think that's a very big possibility. We're also looking at sort of other metabolic sort of pathways that feed into sort of the glutathione sort of synthesis pathway, like the there's a methionine pathway that feeds into the glutathione synthesis pathway. For example, we're looking into that in terms of inhibition of both or both synthesis of cysteine and methionine. I think may potentiate it's a possibility of potentiating apoptosis. So I think there's a this is sort of the whole sort of metabolic pathway that goes through the cytoplasmic component and the mitochondrial component, and there's a potential that a lot of these other pathways may feed into this. Exactly how it's regulated and what's driving what, because there's a lot. Seems like there's going to be a lot of biofeedback mechanisms that occur. So I think that's still to be to delineated in terms of which pathways are going to be important.
Perfect. Thank you. Dr. Curry, we have one more question. Do you want? I think it's for Paul, I believe.
Yeah, I think this one's for me.
Yeah.
Yeah, explaining the co-culture assay. So just a quick explanation would be, so you adhere the cells, you add the cells to microscope slides essentially, and just let them adhere by themselves overnight in the same kind of dish, and then a few hours later or the next morning you add the media. So you have essentially two microscope slides. One you'll treat as the irradiated cells, the other as the bystander. Then you perform the irradiation, and then after you've irradiated, you kind of detach these the cells from the irradiated and the bystander slide, and then you resave them and then measure the clingenic survival. So there is a bit of noise because of the whole reseeding thing you have to do, but that's the only way we could do it. Well, the only way we were able to do it.
Perfect. Thank you.
That answer first.
I would have another question for Dr. Chang with the. I was wondering, because you had one slide where you show several of the pathways, and I understand it's probably going to be a balance between all the different cellular death, which one's going to take over at some point. But also, like we talk about radio resistance, and I could not help thinking that NRF2 pathway is going to have to be kicked in at some point. And to what extent, because especially because NRF2 is going to control the production of the glutathione as well. So how does that fit into this proposed mechanisms that we have for ferroptosis, and then at which point you think that which pathway can take over?
Sure. My lab hasn't looked at NFR2, but I think Boyi Yang's group at MD Anderson looked into it, and also KEP1. I think NFR2 is upstream of KEP1, I believe, and that sort of controls sort of the different oxidative stress pathways within the cell. So I think if I believe if you not, I think those pathways, I'm just trying to remember what their data was, but I think that they definitely affect the ferroptotic pathway. I think if you inhibit KEP1, you do get increased ferroptosis. I have to remember the data, but those pathways are, I think anything that sort of affects sort of the balance of oxidative stress pathways are definitely going to tip potentially, the ferroptotic pathways. And exactly, is it just a fine, are there regulators like NFR2, or if there's, how is it just a sort of a fine balance of the redox pathways, and what are the triggers between, apoptotic pathways and ferroptotic pathways? I think that's going to be a big question, and it's still unknown. There's some suggestion that maybe that P53, may, act in some way, but it's still unclear.
So with the same context, I'm going to bring back again the sphingomyelin, because there was also a paper that showed that the acid sphingomyelinase can be important for ferrolytic peroxidation. So that's why, I see that you cannot really affect one without affecting the other one. At some point, there's going to be some crosstalk, I think, between everything that reactive oxygen species can affect, and then it's probably going to be a balance of how much reactive oxygen species a cell can tolerate that would be, pushing toward one or the other. So I'm just trying, because I know you're looking at which type of lipids, but was the sphingomyelinase pathway part of the pathway that you were looking at?
So is that a question for me? Or
yeah, it's for you. It's for you.
Oh,
yes.
okay. We haven't looked at on sphingomyelin in terms of sort of yeah, that's a good question. We haven't looked at the sort of other cell death pathways besides sort of looking at the difference in apoptosis and ferroptosis. But it's interesting, exactly. Does it drop? There's the lysosomal, necrotic pathway. I think that's it is like, I guess the question is all these pathways just acting as sort of a fine balancing act of redox oxidative stress pathways, or is there actual trigger? Is there a molecular trigger that actually flips one to the other? That's I don't think anybody's that hasn't been found yet.
It's going to be very interesting to sort out.
Yeah, absolutely.
So, yes, absolutely. Yeah, we have more questions in the box. Dr. Patel, do you want to go through those? I think they're more
questions for me.
too. Yeah, for you. You're very popular.
Yeah. I'll go through these here. So have you ever looked into double-strand DNAs that cause activation of C-casting? What resources do they come from, mitochondrial or nuclear DNA? I'm not sure exactly, I haven't looked into it, but all I know about C-gas is that I don't think it's all that picky about which kind of double-strand DNA fragments it detects. So I don't think it's too picky about the length of the double-strand DNA fragment or anything like that. I'm not really too sure. That's all I can really say for the first question. Second question, is there any crosstalk between C-casting and other bystander signaling? So does that mean about other kind of components that could be causing the bystander response? So yeah, I'm not sure if C-gas sting pathway is explaining the entirety of the bystander effect. There could be other mechanisms in there. I did do the assay where I added the G10 sting agonist, and if sting activation was the only component of the bystander effect, you maybe would expect a differential response between the parental and the knockdown cell, but I didn't really see that. So that's maybe suggesting that there are other components to the bystander response there. I hope that answers those questions. Thanks. Let me know if you have any follow-up. From Wisdom Kate, what would you say is the source of the DNA you obtain in the media? Would it be cytoplasmic, nuclear, mitochondrial DNA, or DNA generated as a result of cell death, that is, by dying cells? And secondly, do you have any, it keeps jumping around here, method in mind that would allow you to isolate and quantify the DNA in your media? I haven't been able to pinpoint exactly what the source of the DNA is, whether it's cytoplasmic, nuclear, or mitochondrial. All I'm really doing, so the next assays I planned would be kind of immunofluorescence staining for double-strand DNA. But again, the antibody I'm using there, it'll just highlight up anything in the cytoplasm, any double-strand DNA in the cytoplasm. I'm not sure exactly where the source is. And yeah, something I'm planning to do is an assay that would allow me to quantify the DNA in the media. So there's a double-strand DNA quantification assay I have planned. So I was maybe planning to look at different time points and seeing how the double-strand DNA levels change with time post-radiation. So maybe that would give some kind of insight into the source, depending on what time it appears there. And a final question, do you foresee there might be a C-gas dependent INF response driving the bystander effect? For example, damps produced by cells following cell death. Yeah, they're C-gas independent INF response. Yeah, C-gas sting, I'd be surprised if it was the only kind of mediator of the bystander response. Again, going back to the G10, the assay where I added the G10 sting agonist, there wasn't a differential response there. So it's possible that cell death is driven by other factors too. Yeah, thanks for all the questions. I hope I answered them satisfactorily. If I didn't, just send me an email or send me a message. Thanks.
Thank you, Paul.
I would have a question for Sylvain Perrodon. Are you still there? Hi, bonjour.
Bonjour.
So when I look at your data, I could not help thinking about P10, because P10 is going to have probably a very similar effect as what you're seeing. And could you just comment of the similarity or the differences that could happen, you think?
Yeah, so P10 is mostly inhibitor of the PI3K AKT activation pathway. And from what I found with my data is that COSY would be actually an activator of the PI3K pathway. So they would be kind of a balance between each other. What we have found is that COSY can directly bind to the regulatory subunit of PI3K. And what I'm working on right now is to show that this interaction leads to the activation of P110 alpha, and then follow down to the activation of the pathway and phosphorylation of AKT and mTOR. But I think there is a balance between the effect of COSY on the PI3K and the effect of P10 for the modification of the phospholipids.
Yeah, what I had in mind is more when P10 is mutated. When you have a P10 mutation, then you would result in activation to the PI3K. So that's going to be
correct.
Probably the same thing that you see. So do you know if there would be like super amplification, if you have overexpression of the COSY in the P10 negative background?
So Maybe. I didn't look precisely to this. What I can tell you is that the two cell lines that we used are P10 wild type, but one of them is PI3K CA mutated. And comparing these two cell lines, we actually end up to observe the same effect at the end when we play with the level of COSY. So it seems that even if P110 alpha is mutated, we still can slow down the activation of the pathway if we decrease the level of COSY. So even with a mutation, which is an hyperactivation mutation, playing with the level of COSY still gives you some leverage to decrease the activation of the pathway after irradiation only, because we don't see any differences when the cells are not irradiated. The doubling time is the same. We have seen that in vitro and in vivo, but it's response to stress where COSY starts to play a role.
So in those panel of patient, I think you have 25 high COSY or non-responder that you could look at. So P10 was not a factor in those non-responder or responder.
That's not something that is routinely checked where I used to work. So we don't have these data for these patients, unfortunately. So that I don't really know. What we know is that 80% of chance will be that APC will be mutated, 50% of chance that P53 will be mutated,
right.
And then BRAF, KRAS, the classic Vogelstein pathway.
Okay. That would be interesting to follow up as a way. So is there a way that we can target COSY to increase radio sensitivity? Is there a drug that we can use that would be specific?
So that's exactly what I'm working on right now.
Okay.
The main problem right now is that the effect of COSY that we demonstrated with my team is a non-canonical effect, because the canonical role of COSY is to participate to the de novo synthesis of CoA. And if we use an inhibitor for this enzyme, even the normal tissue will be very impacted. So what I'm trying to work on right now is to do the crystallization of the interaction sites between COSY and PI3K and try to find a small molecule inhibitor that would
disrupt the protein-protein interaction.
Binding. So the enzyme will still have the canonical effect for the normal tissue, but not for the
activation of the pathway. Yes.
Thank you.
Good.
Can I ask Dr. Chang a question? I can't figure out how to type into the Q&A.
Sure.
Dr. Chang, I'm wondering, did you see a dependence on iron stores and sensitivity to ferroptosis or the ability to sensitize to radiation? And if so, do you know what the source of the iron is within the cell?
So we look superficially at iron dependency. It looks like it is somewhat dependent, but it's not. Other people have looked at it. If you eliminate intracellular iron, labile iron, the effects of ferroptosis does decrease. But there's a way around. I think there are pathways probably around it that it doesn't absolutely require iron. I think it enhances sort of the liver peroxidation, the Fenton reactions that occur. And so we looked at it, and also there's probably a transferrin receptor, which brings in sort of the iron 3+ into the cell that transfers to more labile iron. Looks like it's one of the probably transporters that brings in the iron that increases sort of Fenton reactions that occur for liver peroxidation. Is it absolutely required? It's unclear if it's absolutely required or not, though. It does enhance.
Okay, do we have any more questions for our speakers? Or do they have questions for each other? That's fine. This is good. I like that. Yep.
Can I ask a quick question to Dr. Ferdon? So you look really strong COSY expression and the radio resistant in human samples. Did you look at the expression changes post-radiation?
Yes.
And what was those changes?
So, well, very good question. We actually tried to irradiate different types of cells at different doses and looked for the protein expression of COSY. And there is absolutely no changes, whatever the dose we are using, when we look at the protein expression in the next 24 hours after irradiation. And I also looked on kinetics from short time after irradiation, 30 minutes, up to 48 hours, and we haven't seen any differences. So the expression of COSY protein is not affected by irradiation, either of the dose or the time.
Got it. Thank you.
Yeah, I think our session has to come to a close now, right? I think 3:30 was our time allocated. So thank you everybody for speaking and joining the session. And I think it was very productive, very interesting, and I'm very glad to see that we have a very strong follow-up, young people coming behind us, the older people. So that's good to see. I think the field is strong and a lot of good things coming up. So enjoy the rest of the meeting, and thank you again for your participation.
Thank you, everyone.
Thank you. Thank you, everyone.
And thank you for my co-chair, Dr. Patel. Thank you.
Thank you.
Thank you.